This is lecture 13 on the topic of plant signals. This lecture is separated into three components, and this is part 3. Please ensure that you watch part 1 and part 2 before commencing with part 3. This lecture is part of the subject Plant Physiology, which is a component of the Bachelor of Agriculture and Technology, a degree offered jointly by both Melbourne Polytechnic and La Trobe University. Please visit our website at www.melbournepolytechnic.edu.au for further information on this course and other courses that we offer. My name is Dr. Nikki Cooley. In part 3 we are going to explore ethylene. We are going to look specifically at its role and function. This will be followed by a section on abscisic acid or ABA. Again, we will examine the role and function. And finally, we will finish on the group, the Brassinosteroids. We will look at the role and function of these hormones as well. This lecture will be concluded with a summary on plant defense. So let us start with the plant hormone ethylene. Ethylene is a very light compound. It consists of 2 carbon atoms, which are presented on the slide as the 2 black balls, with 4 hydrogen atoms, which are presented by the 4 blue balls. Ethylene is a gas. During the 19th century, when coal gas was used in street lamps, it was observed that nearby trees defoliated more extensively than other trees in the area. In 1901, a botany student in Russia observed that dark-grown peas exhibited symptoms later called the triple response. That is, they had reduced stem elongation, increased lateral growth, and abnormal horizontal growth. Ethylene from coal gas in the lab was responsible for these responses. Almost all parts of the plant can produce ethylene. Production increases during leaf abscission, flower senescence, fruit ripening, wounding and physiological stress, including flooding, disease, temperature and drought stress. The amino acid methanone is the precursor of ethylene. The major enzymes in this pathway are ACC synthase and ACC oxidase. The ethylene biosynthesis starts with induction of ethylene biosynthesis. The figure on the screen illustrates some of our understanding to date into the insights of this complicated process. You will not be required to remember this process, but only appreciate the complications. After ethylene biosynthesis has occurred, downstream ethylene sensing and signaling pathways occur. Again, these are shown on the slide. In a recent paper published in 2006, ethylene biosynthesis in rice was illustrated. For the level of this subject, you will not need to know the intricacies of this pathway. Only that there are many steps and that there are many intermediates. Ethylene is the hormone that promotes ripening in some fruit. Ripening is the softening due to enzyme breakdown of the cell walls, starch hydrolysis, starch accumulation, disappearance of some acids and phenolics, including the group tannins. Dispersal by ingestion is where ripening also involves the accumulation of brightly coloured anthocyanins or carotenoids. Ripening means that the seeds are ready for dispersal. Fruits that ripen in response to ethylene exhibit a respiratory rise called a climacteric before ripening. The figure on the slide in banana illustrates ripening characterized by climacteric rise in respiration of carbon dioxide, which precedes by an increase of ethylene. Fruits can be separated into 2 groups, those that are climacteric and those that aren't. The table on the slide shows you some examples of each of these groups. Ethylene induces root growth. It has been shown that ethylene can induce advantageous root formation in leaves, stems, flower stems and other roots. For example, vegetative stem cuttings from tomato and petunia. Ethylene also promotes root hair formation, as shown on the illustration in the slide, in lettuce seedlings. Ethylene is a hormone that can enhance leaf senescence. For example, exogenesis application of ethylene accelerates senescence. Inhibitors of ethylene retard leaf and flower senescence. Carnations in silver thiosulfate, or STS for short, is an inhibitor of ethylene action, and it results in inhibition of senescence. Again, understanding the function and mode of action of ethylene in plants has been very useful from the terms of the commercial agricultural perspective. Ethylene is one of the most widely used hormones in agriculture. Effron, discovered in 1960s, is sprayed in solution to hasten the ripening of apple and tomato. It synchronized flowering and fruit set in pineapple. It induced fruit thinning in cotton, cherry and walnut. It is responsible for degreening of citrus fruits. It prevents self-pollination and increases yield, and it can promote lateral growth. The ethylene inhibitor, ethyl block, is used to extend the shelf life of various fruits. The next hormone we are going to cover in this lecture is abscisic acid, or ABA. On the slide you will see a molecular structure and a modelled structure by the Nutritional Immunology and Molecular Medicine Laboratory. ABA is a phytohormone that serves as the prime signal in response of plants to environmental stresses imposed by cold, drought and high levels of salt, as stated by Christemin et al. In their 2006 paper. ABA was originally discovered in leaves from sycamore trees and named dorman. ABA is an important plant hormone that regulates growth and stomata closure, seed maturation and dormancy. ABA is synthesized in the chloroplasts and the other plastids via the terpenoid pathway. ABA is transported via the xylem and the phloem, but is much more abundant in the phloem. ABA biosynthesis, like many of the hormone pathways, is complicated and intricate, and it has many intermediates. From the perspective of this subject, you only need to understand that it has many intermediates. ABA is one of the components that is responsible for the closure of stomata. ABA concentrations in leaves can increase up to 50-fold times under drought conditions. Redistribution of ABA is very effective in causing stomata to close. Its accumulation in water-stressed leaves plays an important role in the reduction of water loss through transpiration. On the figure on the slide, produced by Christemin and Gill in their 2009 paper on ABA, they demonstrate how the compound ABA is involved in the opening and closing. On the right-hand side of the picture are some of the genetic pathways involved in this process. Let us examine the process of ABA and stomata closure in more detail. ABA binds to receptors. The ABA binding induces the formation of reactive oxygen species, or ROS, and this in turn activates calcium channels on the plasma membrane. The influence of the calcium interacts with intercellular calcium transients and promotes further release of calcium from the vacuoles. ABA stimulate no production and no increases of the CADPR levels. ABA does increase the INSP3 levels via a signaling pathway that induces S1P, heterotrimeric G proteins, and phospholipidase C and D. The rise in the CADPR and INSP3 activates additional calcium channels on the tonoplast, releasing more calcium from the vacuoles. The rise in the intracellular calcium blocks potassium channels on the plasma membrane. The rise in the intracellular calcium promotes the opening of chlorine ion channels on the plasma membrane, causing depolarization. The plasma membrane protein pump is inhibited by the ABA-induced increase in cytosolic calcium and a rise in the intracellular pH, further depolarizing the membrane. Membrane depolarization activates potassium plus ions out of the channels and onto the plasma membrane. Potassium ions, to be released across the plasma membrane, are released first from the vacuoles in the cytosol. In summary, this process is an elegant process that involves not only hormone signaling, calcium and potassium and chloride channels, and ATP in the form of energy. Stomata close due to the net effect of the loss of potassium ions, chloride ions, and the subsequent osmotic loss of water. The surface area of the guard cell plasma membrane might contract by as much as 50%. The loss of turgor induces closure of stomata to prevent further water loss by evaporation. And this brings us to the last group of hormones we are going to study in this lecture, the brassinosteroids. On the screen you will see a typical structure of a brassinosteroid. There are also images of Arabidopsis and mutations involving brassinosteroids. The brassinosteroids, or BR for short, are a group of steroid hormones that play a role in cell division and cell elongation in stems and roots, reproductive development, leaf senescence, and some stress responses. The early studies in the 1960s showed that the greatest growth stimulating activity was found in extracts of pollen from rape plant, shown to increase total biomass and seed yield. Therefore, the US Department of Agriculture provided funding for laboratories to purify and identify the compound. Approximately 60 related brassinosteroids have since been isolated. They are synthesized via the terpenoid pathway. In a publication in the Brazilian Journal of Plant Physiology in 2002, a review article was written on the brassinosteroid pathway. The pathway in that review is presented on the slide. As with all other biosynthesis of plant hormones, this pathway is complex and involves many intermediate steps. Small-scale studies have shown that brassinosteroids can increase bean crop yield by 45%, enhance leaf weight of lettuce by 25%, increase yield of rice, barley, wheat and lentils, promote potato tuber growth, increase resistance to infections, and enhance tomato fruit set. While large-scale studies in Japan, China, Korea, Russia have shown variable results, minimal effect on crops grown under optimal conditions, or dramatic effects on crops grown under stress conditions. BRs are a useful aid for plant propagation. For example, treating wooding cuttings enhances the root response. Micropropagation of cassava and pineapple has been improved by BR treatment. Please read Chapter 13 on the topic of secondary metabolites and plant defense. Read these in sections and insert your notes from the reading here. Please watch the following video by Mr. Anderson on plant control. That brings us to the end of the topic on plant signals and plant hormones. In summary, you would have now watched 3 lectures, watched a number of video resources, and read several chapters in the recommended textbook. You will have learnt about the main plant hormone groups and some important mechanisms, how plant hormones can control these mechanisms, and therefore a plant response to a typical environment or climate. You will have learned that hormones are complex and that they can work together. They act as a finite detail control for plants. That brings us to the end of part 3 of this lecture and this topic.